Hello, everybody, and welcome to Physiology of Activity. I feel as if I'm in some kind of weird vortex from hell that I am back recording lectures in this format, so I can only imagine how you guys feel. Um, Bear with me. We're going to make the best of the situation. If you happen to be listening to this lecture prior to your first lab day, I am going to do kind of an introduction to the course on the first day of lab. So I'm not going to put it in here because I think describing how a course is formatted works much better in person where you can ask questions. So I'm going to jump right into the lecture here and just know that a description of the class is coming if, like I said, you happen to be listening to this first. All right. This is such a fun class. Physiology of activity is kind of my upper class. Pathophysiology, I will admit, is a bit of a downer because it's kind of all the what goes wrong in the body and what happens with disease. And I find it fascinating, and I really love the class because I love physiology, and I love the appreciation that happens when one small part of a system fails to work properly and the ripple effects that has through our whole body. I find that incredibly fascinating, and how the medical interventions, the surgeries, the drugs that we have designed to counter this, I find fascinating. But the class is, I would admit, a bit of a downer because it's all about disease. This class is the opposite. This class is what we can do about it as physical therapists. And I think it's incredibly exciting. This is actually the whole reason that I became a physical therapist. Was I liked medicine. When you like science and you think the body's cool, you kind of debate becoming a doctor or a nurse, or like whatever you think you might do in the medical field. But the ability of what we can do to help our bodies naturally, just through the tools of what your body already has to help make life better, was so exciting to me. I thought that was cooler than any drug, any surgery out there. And that is the reason I went into physical therapy, because you can heal the body from within if you just know what is the right thing to do with your body. I think that is awesome. So this class I'm very excited about. I hope to bring you on this journey. The way the course is built. So this is the exercise physiology content of the PT curriculum. Now, I am very aware that about 60% of you, maybe even a little more now, have an undergraduate degree in exercise physiology, and that 30 to 40% of you do not. Okay, and so that is kind of an underlying tension in this class, because this class is exercise physiology. I have designed the class in a way that hopefully this content will be presented to you in a very PT specific way and through the lens of the patient populations that you will be treating. So for those of you with exercise physiology backgrounds, I'm hoping this is going to take this information in a very PT applied way so that you feel like you're getting usable content that is building upon your foundational knowledge. It's important to know that if the amount of information that's in an undergraduate major of exercise physiology is a lot more information. And this class is designed for what parts of exercise physiology does a physical therapist need to know to be successful. So there's definitely going to be areas of this class where those who have an undergrad major went into a much more detailed place with that information, because it was important. If you're going to graduate as with an exercise physiology undergrad, you need to know everything down to the nitty-gritty. We are now at a graduate level of what about that big, huge field, exercise physiology, is the most relevant for a physical therapist to have in their knowledge bank to prescribe exercise. And it works well, I think, as a new class, because a lot of what exercise physiology is a study of how to take the body to its extremes, or its how can you best make the system work. And you kind of, a lot of the research in exercise physiology is on like normals and how you make them improve. How you get to the athletes that can jump higher than anybody else and have VO2 max is greater than everybody else. And that's super exciting, but that's not a lot of what physical therapists do. We're very rarely taking normal able-bodied people and making them extraordinary. It's much more likely we're taking someone who is below normal, who has lost physical ability, who has severe muscular atrophy, who has a failure of the respiratory system. And we are trying to apply the concepts of exercise physiology to bring them up, right? To raise up their abilities using exercise as medicine. So it really is a different way in some respects. I'm sure you've done that in classes in exercise phys too, but it is a different way of looking at exercise as a means of trying to maximize a system that has a fault in it, that has something that isn't working right, and how can exercise still be appropriately prescribed and used in a way that can make your patients' lives better. For those of you who are not exercise physiology majors, you do not need to be an exercise physiology undergrad to be successful in this course or to be a good physical therapist. It is great for all you guys who have that extra knowledge. It's an enriching thing, just like each and every one of your undergrads gives each of you kind of a unique, enriched background. But you don't need to have all of that to be a good PT. And I absolutely know that you don't need that undergrad to be able to be successful in this class. I will introduce concepts and terms that are new to a non-ex-physser as we go along. And hopefully it is presented in a way that is interesting for both groups. Phizzers, non-ex-phizzers. All right, we are going to jump in first. The first lecture of the semester is on skeletal muscle physiology. I think this is the best place to start because we are really the muscle experts in the medical field. You guys get so much more education on skeletal muscle than any other profession. This is our expertise, and a full, deep understanding of how the muscle works, how we communicate with that muscle to get the sort of force generation that we're looking for is absolutely key to being able to prescribe exercise as an intervention. So we're going to start there and then build through all the different systems of the body as we go through different special populations that you will encounter in the time of your practice. So exercise as medicine is one of the key elements of being a physical therapist. And this class is the physiology of the exercise prescription. And I lead with this slide because exercise is tricky, because almost everybody knows what exercise is, right? This is not a new concept for anyone. We could stop someone on the street and ask them what they know what exercise is, and they could probably give you a pretty decent explanation of what it is. We pretty much all know exercise is good for us in some way, shape, or form. People will be able to explain that in different degrees. And most of you probably are walking into this class with a pretty good idea of what the general recommendation for exercise is. If we were in class, I would have you guys write this down. So if you want to quiz yourself, pause the video right now and write down just on the corner of your notes what you think is the appropriate prescription for exercise. What's the general recommendation to the public on exercise? Hit pause and write it down for me for a second. Hopefully you restarted the video at some point because I only told you to hit pause. I didn't tell you to restart it once you wrote it down. That was implied. I hope you guys are back with me. Okay, what did you write down? I think a lot of people, even non-PT expert people, would probably say something around 150 minutes a week. Some of you might have gone into a little more detail. 150 minutes a week of moderate intensity exercise. Some of you might have known that if you up the intensity, you can decrease the time. So 75 minutes of vigorous exercise a week. So you're coming to this knowledge, you kind of know what exercise is. We kind of know that exercise is good. We've kind of a general feeling of how much we should do. But we really need to think of exercise in a much more specific manner than these generalizations that a lot of the general public even have. Because as PTs, we prescribe exercise. And I really want you guys to think about that just the same way that a medical doctor would think about prescribing a pill. A medical doctor would never just hand a bottle of pills of all different sizes to a patient, throw it at them, and say, this is good for you. Take this regularly. See you in a month or see you in a year, right? Every pill would be the right dosage, right? The right amount would be in each pill. So that would tell the patient how much of the medicine to take. There would be an explanation of how often they should take that pill. They would give an explanation of what kind of a medicine they're prescribing, right? And we prescribe very specific exercise, from strength training to aerobic endurance activities, flexibility exercises, and each one of those has a very specific goal. I want you guys to really start filing away this thought that when you prescribe exercise, it should be in the same detail that a pill bottle would be given to someone. What's in here? Why is it good for this patient? Why is this specific pill being prescribed? How much are they taking? How often are they taking? And be able to communicate that in a way that the patient understands. Okay, let's start with a case. Our first case is going to be a 58-year-old male with unremarkable medical history who presents to an outpatient orthopedic clinic for chronic upper back and neck pain. Very, very typical patient. All right, here is this patient. And just using eyeballometrics here, we can't put our hands on this patient. What do you see? Remember, he has chronic upper back and neck pain. Just looking at his body, what are some things that are notable to you? If you would like to, because normally I would do this in a discussion, you can hit pause and write down your observations. Write down your observations and we'll come back to them. All right, so if you're logged back in or you just listened the whole time, let's talk about what we found. So what are some things that we see? Hopefully what jumps out at you is some pretty poor posture, right? He has forward shoulders. He has definitely a forward head. You can see this exaggerated upper cervical extension to keep his head forward with this poor upper posture. An increased thoracic kyphosis. I see some mild scapular winging. So when you're seeing those things, right, you're starting to kind of make a list of the problems that you might be underlying this patient that could be leading to this chronic upper back and neck pain. So let's go through a few of those. Some scapular winging. What could be a cause of scapular winging? If you've got really poor serratus anterior strength, you're going to see those tips starting to poke out. Forward shoulders. What's creating the environment where he's standing there naturally with these forward shoulders? I would guess if I'd get my hands in there, I'd feel some pretty tight pec musculature, pretty weak scapular stabilizers, right? That's allowing the shoulders to be dominated by those short muscles in the front from the pec. Increased thoracic kyphosis. Yeah, I'd expect some pretty tight cervical extensors and weak elongated cervical flexors. So if this person was in your clinic walking in, what would you do? Well, you'd use your test and measure skills, right? You'd measure the strength of the muscles that are involved. You'd measure flexibility of the muscles that you think might be contributing to the problem. This semester and next semester, you're going to dive into ortho, right? After you collect your evidence and test and measures, orthopedics is going to give you the skills to diagnose the musculoskeletal injury that's associated with the poor posture and help you decide what do you do next. But absolutely, for this patient, one of the next steps would be to prescribe exercise. Right? This patient would have great potential for improvement with a appropriately designed exercise prescription. What is going to be the target of that exercise? This patient's exercise prescription is going to target skeletal muscle. You are going to prescribe exercise to make a change in serratus anterior strength, scapular stabilizer strength, maybe even cervical flexion strength, flexibility, length of the pec muscles. These are going to be the targets of your exercise prescription. So if skeletal muscle is going to be your target, we have to understand how the skeletal muscle is arranged, how it's structured, how it works, to know how we can influence it to make a change in our patient. So let's talk about muscle. The kind of middle of your notes, there's just some interesting factoids, obviously not memorizable for a test, but important for us to have a feel about, right? We have over 600 muscles in the body. About 45 to 60%, based on your leanness, would be the percentage of your total body weight that's skeletal muscle. So as body fat would go up, the percentage of lean would go down, right? So the percentage of your total body weight would go down. Muscle has a ton of water in it. As you can see, 70% of it is water. So you should really think of skeletal muscle as almost a water reservoir. In your total body water, about 5 to 8 liters of that water is in the blood. So if we are dry in the blood, right? If we are depleted of water, the body's going to take it from the muscle because that's a water reservoir. So to maintain those high liters of water necessary for our circulatory system, if we're not drinking enough, if we're dehydrated, the body will steal it from muscle, and that'll impact muscle function. So something to keep in mind, dehydrated patients are going to have affected skeletal muscle function. About 20% of the muscle is protein, about 12% of that is contractile, or of that 12% is contractile. And then there's a whole bunch of enzymes and channels, and this is what makes the energy that allows the muscle to work. So I think B in your notes is pretty obvious. The functional requirements of skeletal muscle is it needs to contract and generate a huge range of tension and forces. That my example I always love is when one of my little nephews was like 1.5, and I was trying to get him to pull grapes off of a stem, like a whole big pile of grapes. And he couldn't do it. This kid was strong, but if he held on to the grape, he hadn't yet figured out how he would either squash the grape or he couldn't pull it off. Like, to keep his hand not squeezing hard enough to squash the grape, he couldn't generate enough force to pull it off the vine because he was too little and he hadn't coordinated that degradation of force. Now you don't even think about it, right? You can hold a grape very lightly in your fingers and yet still generate the force to pull it off the vine. That's amazing that our body can do that, and we completely take it for granted. And that's just a silly example. You guys will see much, much more vital, critical examples of that in your patients, that when there is a decreased ability to create that variation from fine motor to gross performance of strength, it just has a huge impact on function. So we had to be able to alter tension and force in very small increments, and we have to be able to do this over and over and over again. Some of our muscles, right, need to be able to do it for hours on end, our postural muscles, or some muscles that are engaged in extreme power, like our quads, our explosive energy, had to be able to create this force so rapidly. I point this out and I say it out loud, even though you guys know this stuff, because I hope you never stop being just completely amazed by this system. It is extraordinary what the range that we possess within our skeletal muscle system and what it's capable of doing. So that all comes together to allow the functions of skeletal muscle at the bottom of your notes. Locomotion gives us all of our posture, support and protection of internal organs. Huge role in bioenergetic homeostasis, maintaining that energy and heat. It assists venous return and a key role in thermogenesis. Let's dive into the skeletal muscle itself. So the whole muscle is, you can see here up at the top, consists of many, many individual muscle fibers. So I'll just kind of cartoon along because I do better, as you guys know, when I draw. So whole muscle is many, many muscle fibers together, and the whole thing is wrapped in an epimysium that surrounds the entire muscle belly, and it's what separates one muscle from other muscles. So the outermost layer, that is why when you guys did gross dissection, right, you could just kind of slide your fingers between whole muscles and you could easily feel the muscle belly together because it's wrapped in epimysium. Within that skeletal muscle, you see these bindings of a fascicle, and that's a bundle of skeletal muscles. So you kind of have like a bundle here, and it's a bundle of many different muscle fibers all together, and that is seen here in the slide form. So this is a fascicle, and each fascicle is wrapped in perimysium, and that divides the muscles into groups of muscle fibers. It also provides a really important conduit for blood vessels and nerves to travel along with the muscle. So within each one of these is the individual muscle fiber. And the muscle fiber is synonymous with the muscle cell or the myocyte. For some reason, it always took me a while. Muscle fiber didn't feel like the same as a cell to me, but that is the individual muscle cell. A skeletal muscle cell is very unique in that it has multiple nuclei, but this is truly the cell. It extends kind of the whole length of the muscle. Each individual muscle fiber is wrapped in endomysium. It basically is the wrapping immediately outside the sarcolemma. This is an important site of metabolic exchange between the muscle fiber and the capillaries. And then each muscle fiber, so here is the muscle fiber here. If you look inside, you can see all these kind of strands maybe inside of each individual muscle fiber, and those are the myofibrils. So the myofibril is the basic rod-like unit of the muscle cell that's made up of our thick and thin myofilaments. So each muscle fiber contains many of these tiny strands, and these are somewhere between like 1 to 2 micrometer. So really tiny little strands that are all contained within the cellular membrane of the muscle cell, because the muscle fiber is the individual cell of the skeletal muscle. The myofibril is consisted of many myofilaments. And again, this picture kind of zooms in even further so you can see the myofilaments. Myofilaments are thin and thick fibers that are the basic organizational unit of the skeletal muscle fiber. So the myofibril is made up of these myofilaments. And let's talk about each one with a little more detail. So in this picture at the top, the red line here is the thin filament, which in skeletal muscle is primarily actin. So actin is the thin filament. As you can see in your notes, it's about 7 nanometers in diameter. And this is here on the cartoon, the zoomed in picture of actin. This little kind of beaded rope is the actin. In this cartoon, you can see the yellow color on the cartoon is meant to be the active sites of actin, which are covered by this kind of ribbon-like structure called tropomyosin. And the tropomyosin is bound with, let's see, where's the best place you can see it? Kind of these little brown balls here is the troponin. Those are going to be some keywords when we start talking about skeletal muscle function. The thick filament in the middle is this purple, and that is primarily myosin. Myosin is called the thick filament because it's got a thicker diameter, about 15 nanometer in diameter. It's truly several hundred molecules of myosin that are all wrapped up together, and it creates this big thick filament. So if you zoom in, you can see the individual, all those little myosin kind of roped together, and the little bumps popping up here are all the little myosin heads. So if you zoomed in all the way down to just one myosin molecule, you could see the myosin tail, and then it has the myosin head at the tip. The other structure I want to draw your attention to is kind of the green squirmy line on here, and that is the elastic component, which is titin. Titin is a really, it's the largest known protein, and it's often drawn like a spring or a coil because it has this really important ability in the muscle to store energy and facilitate a contraction, because it has this ability to stretch. And that will come into play when we start talking about the functional properties of skeletal muscle. So these myofilaments within an individual muscle fiber are organized in a very systematic way. And this organization of the myofilaments, is the sarcomere. The sarcomere is the basic functional unit of the skeletal muscle, and it is a repeated structural element all the way along the muscle. This picture is in your notes to give you an illustration of how the muscle is arranged within the whole unit of the muscle attached to the bone. So to orient you to this picture, this is a picture of a muscle attached to bone. So on either end, you have the bone, and muscle, as you guys obviously know, is attached to the bone via the tendon, which is listed on either side here. And this whole central section is meant to illustrate the properties of skeletal muscle. So muscle is composed of both contractile and non-contractile elements. When you're looking at this figure, you can see non-contractile elements are the extracellular connective tissue. So the paramycium, the epimycium, the tendon, those are all connective tissues that are part of the skeletal muscle, but do not have any contractile properties. Titin would be listed there. That is that big molecular spring that we just talked about in the previous slide. The non-contractile properties of skeletal muscle may align in series or in parallel. So you can see that, tendon is in series with titin, whereas the extracellular connective tissue of the membranes that surround the muscle would be in parallel. So the non-contractile properties are paired with the contractile element of the muscle, and that is the sarcomere seen here at the bottom. So the sarcomere is the functional unit of the muscle fiber, and it contains the contractile elements. A sarcomere is a basic repeating unit that is between two Z lines. So here on either end are the Z lines, kind of what anchors the sarcomere to its kind of front and back. The organization of these repeating sarcomeres is so consistent. This is actually why skeletal muscle is called striated skeletal muscle, because if you zoom in under a microscope, you can see these repeated bands, these repeated functional units of sarcomeres. And as you can see in the picture, the subunits are in almost nearly perfect alignment with those kind of next to each other. They're lined up very, very carefully. And you can see the elements of the sarcomere here on the electron micrograph. So this first area is called the A band, and you can see that here is this darker area, a kind of a dark band right in the center of the sarcomere. Here, in case you're missing it. The A band is the thick filaments, and the thick filaments, again, are largely the myosin. So this is that central border, the central area of the sarcomere that has the myosin filaments. On either side of that, you will see the I band. The I band is this lighter area that represents the actin. So the I band you can see, so here's I band on either side of the A band. And then the last thing we mentioned in the previous slide is the Z line. The Z line, if you can see here, let's add them to this guy. Here's the Z line on this sarcomere, Z line on this sarcomere. And that is what truly anchors the sarcomere. Actin and titin connect to the Z disc at the Z line, and that creates the borders of an individual sarcomere. Now that we have the overall framework of how the structures within the muscle are organized, let's first talk about how we communicate to the muscle to stimulate a contraction, and then we'll go through and talk about how that contraction actually happens. The ability of us to connect our nervous system or our brain, deciding we want a muscle to move, to the actual functional properties of skeletal muscle is called excitation-contraction coupling. This sounds potentially more intimidating than it is, in that it's just the connection between the neural impulse and the ability of the skeletal muscle to contract. So it is the link between the chemical and electrical signaling between brain and muscle. So I have this picture available to you. I do have space in your notes for you to draw this process out. I think this is one of those that works better if you kind of see it in real time and can draw it along with me. So I'm going to kind of draw what was in that cartoon here for you today. So first we're going to have the motor nerve coming down. And this is going to be my motor nerve. Just going to shrink that guy up a little bit. So at the end of the motor nerve, there are these presynaptic vesicles, kind of little stored pockets of neurotransmitter. So we have some presynaptic vesicles. And stored within them, ready to use, is the neurotransmitter acetylcholine. So these little red dots in my picture are going to represent acetylcholine, which is I'm just going to give you a little diagram here, ACH, which is the neurotransmitter of the neuromuscular junction. So the motor nerve is sitting here, ready and waiting to interact with the skeletal muscle whenever you need. It's already made its neurotransmitter and it's storing it in these little vesicles. What's really cool about the neuromuscular junction is that the end of the nerve actually burrows down into the sarcolemma. And if you're not familiar, the sarcolemma is just the name of the skeletal muscle cell membrane. So I'm going to draw that here and kind of just squiggle it down because it really does burrow down below the surface into the sarcolemma to be able to communicate with the muscle. So this squiggly line here is the sarcolemma. Oh, is it going to fit? Just barely. And that is the membrane of the skeletal muscle cell. And the membrane does this really cool thing in skeletal muscle. It dives way deep down into the cell in a structure called the T-tubule. And this is really important because it's going to allow activity that would be happening at the cell membrane to get kind of deep down into the muscle. So this, on our picture here, this represents inside the muscle cell, and out here is outside the muscle cell. So this T-tubule dives kind of way down into the cytoplasm of the cell, And that is because it wants to be, I'm going to turn this a different color. It wants to be close to the sarcoplasmic reticulum. And that's just this big tank that sits inside of your skeletal muscle cell. This is your sarcoplasmic reticulum. And it is a big tank of calcium. So I'm going to make calcium green dots in our figure. And if I could draw a million of these without wasting your time, I would tell you, because this thing is chock full of calcium. If I can spell it right. That's supposed to be an I. Calcium. Ready and waiting to dump calcium onto that sarcomere that we just described. So the T-tubule is intimately interrelated with the sarcoplasmic reticulum by these receptors. DHP is the dihydropyridine receptor. I'll write that out down here where it's not going to take space in our picture. Dihydropyridine. And this is one of the coolest receptors. I'll try not to geek out on you guys too much, but it has like physical connections to these ryanodine receptors on the sarcoplasmic reticulum. We'll leave that drawn like this. It's important to know that the DHP or dihydropyridine receptor is a voltage dependent channel, basically, that will open up in response to an electrical potential. And that'll be really key in the communication between the motor nerve and skeletal muscle action. Okay, so what do we have laid out here? We've got the motor nerve at the top. We've got presynaptic vesicles chock full of acetylcholine. One more thing I want to add on is in the sarcolemma, a different color. What color should we make it? Blue. On the sarcolemma, right here in this neuromuscular junction, are a bunch of receptors. So I'm just going to draw some receptors in here. And these guys are nicotinic acetylcholine receptors. Nicotinic ACH receptors. Kind of ready and waiting to respond to acetylcholine. All right, so we have set the framework for the relationship between the the motor nerve and the muscle. Now it comes time to activate this muscle. So your brain decides, I want my bicep to contract, and it sends down an action potential, right? Action potential travels down the motor neuron extremely fast because of our beautiful myelin that allows saltatory conduction down the nerve. So from brain to nerve, although it's a long trip, it goes very fast, and that action potential shows up at the motor end plate, and the action potential causes the synaptic vessels to fuse with the membrane and release their contents. So if you look in your notes, there's some verbiage on the right-hand side that kind of writes out the steps. So we're on step number 2. It says action potential stimulates ACH release in synaptic cleft. That is because these vesicles fuse with the nerve membrane, kind of fuse, and that means that the contents within the vesicles can spill out into the end plate here. So when ACH spills out, well, there's these nicotinic ACH receptors that are just waiting to bind to acetylcholine. So acetylcholine binds to the receptors on the muscle sarcolemma, and the binding of acetylcholine causes an action potential. So the ACH causes a depolarization. I'm just going to do orange lightning bolt is going to be my cartoon version of depolarization. So the binding of ACH causes depolarization of the sarcolemma. And the fancy name for this is a motor end plate potential. Motor end plate potential, or an MEP. But you can think of this depolarization of the end plate as kind of the action potential is now happening on the skeletal muscle membrane. We're having a depolarization and an action potential, and that propagates along the surface of the sarcolemma. You get this depolarization wave that happens. And because the sarcolemma dives down into the cytosol at these T tubules, the action potential dives down along with it because it rolls along the sarcolemma. As it gets down the T tubule, when the depolarization wave hits the dihydropyridine receptor, that is a voltage dependent receptor channel, and that is going to open and activate the ryanodine sensitive receptors on the sarcoplasmic reticulum. So that this physical little connection here, the voltage gated activation of the DHP, is what causes the ryanodine receptor, which is here, the purple lines, to open. When the ryanodine receptors open on the sarcoplasmic reticulum, there is a huge concentration gradient of calcium because there's tons of calcium inside of the sarcoplasmic reticulum and almost no calcium in the cytosol of the muscle fiber. So calcium starts to flood out of the sarcoplasmic reticulum. In this way, the action potential that was generated by the nerve was transmitted across the motor end plate to cause an action potential on the muscle fiber membrane. That action potential on the surface of the muscle fiber triggered the opening of a channel in the sarcoplasmic reticulum, allowing calcium to leak out. Calcium is the important trigger of the cross-bridge cycle, of the ability of the skeletal muscle to contract. Calcium is key to the contraction. So this link between the electrical signaling coming from your brain and the ability of the skeletal muscle to contract is dependent on the release of calcium. That is what's going to allow the physical contraction to take place. Because of that, let's just take a minute to really dive into what calcium does in the skeletal muscle. So when we talked about the structure of the myofibril, we said there were all these myofilaments, thin and thick. So let's first talk about actin, which is that main thin filament. So here is my cartoon picture of actin. And on actin, there are active sites. So I'll make those red circles on here. The red circles are meant to represent the active sites on the actin. And at rest, tropomyosin is, that might be too big. Tropomyosin is this ribbon-like protein that wraps around the actin and covers the active site. So I'm going to draw the tropomyosin on like this. It wraps around and it physically covers the active sites so that they cannot be seen or bound. This is tropomyosin, the ribbon-like protein. Tropomyosin is bound to troponin. And I'm going to draw troponin, kind of disproportionately large for cartoon purposes, as 3 circles because it is really a complex of 3 regulatory proteins. So let's just make this guy. This is our troponin here. And troponin is really 3 separate regulatory proteins. You have troponin C, which is the subunit that has a binding affinity for calcium. So it has a calcium affinity, and that's where it gets its name C. There is troponin T, which has a binding affinity to tropomyosin. That's where it gets its T name. So this is the subunit that holds on to tropomyosin affinity. Here we go. That holds on to tropomyosin. And then there is the subunit I, which I is representative of actin. So this binds to and is drawn to actin. So in the resting state, the active sites are covered by tropomyosin. And the significance of that is because right next to this thin filament, right, are those thick filaments, the myosin, with the myosin head reaching out. So here is the myosin head. And the myosin head would like to bind to the active site on the actin. But if the tropomyosin is covering the active site, there's no connection that can be made. When calcium is released from the sarcoplasmic reticulum through the action of that motor end plate potential opening the ryanodine receptor on the sarcoplasmic reticulum, calcium can bind to tropomyosin C. When tropomyosin C is binding to calcium, there is a conformational shift of troponin that pulls the tropomyosin off of the active site. So the ribbon here gets kind of pulled back and released. So whenever calcium is binding to troponin, the troponin complex will turn and kind of wiggle, and it pulls the tropomyosin off, and the active sites become open and available for myosin to bind. So calcium is key to the muscle contraction, because without calcium, the active sites on the actin are covered and you cannot get a contraction between myosin and actin. When calcium is present, calcium binds to troponin, which pulls tropomyosin off of the active sites and allows myosin to bind. So in the PowerPoint slides, there's cartoon versions of these two things where you can kind of see them in a different orientation, if that maybe works better for you than my cartoon drawing. You can see the actin with the active sites covered by the tropomyosin, and the troponin complex here binding and held in place. When calcium has bound, it pulls the ribbon away, and now you can actually see the active sites, and they would be ready and available to bind to myosin. So I'm going to end this video here right before we get to the power stroke. And this will give you a chance to break up your listening, but you can come back to the next lecture if you are ready. Thanks for your attention. See you soon.